Hello, thank you for the opportunity to present this work. My name is Meridith Kisting, and I'm a research intern at the University of Wisconsin. I'll be presenting this talk titled The Safety and Efficacy of Volumetric Histotripsy Delivery Through the Overlying Gas-Filled Bowel Near the Focal Point in an Ex Vivo Swine Model. These are the disclosures. Histotripsy is an emerging clinical therapy for the treatment of abdominal and pelvic tumors. It's non-invasive, non-thermal, and non-ionizing, and it is focused ultrasound that creates cavitation, which results in mechanical destruction of the cells. There are tissue selective properties based on the tissue that is being targeted. Due to the anatomy, bowel is often present near the targets of ablation in the abdomen and pelvis. There's always concern for bowel perforation and documented cases of this with all thermal ablations. This is an example of an abscess that formed after a bowel perforation from a HIFU treatment of the uterus. Histotripsy is unique in that blood vessels and bile ducts are spared when included in the treatment, as seen in this picture here, this bile duct, but little is known about the effects of histotripsy on bowel. Right now, the safety and efficacy are unknown. So will the bowel be damaged if included in the beam path? And can a treatment be created if bowel is blocking the beam path? The purpose of this study was to evaluate the safety and efficacy of treating through partial blockage by gas-filled small bowel in the therapeutic beam path with a clinical-grade histotripsy therapy system. To study this, we used an ex vivo swine model. So we took the small bowel from healthy swine, inflated it, and placed it in a degassed water bath. This would simulate a scenario where you might have gas blockage, as you definitely would in the pancreas and often could in the case of treating the liver or kidney or even the uterus. We used a clinical grade histotripsy system. This is the same one that was used in the first in human study in the Teresa study. To simulate various clinical scenarios, we varied the distance between the gas-filled bowel and the phantom. We tested this with the bowel touching the phantom or 0 centimeters above, 1 centimeter, and 2 centimeter, as well as a control with no bowel. Additionally, we tested using twice the voltage required to have a therapeutic effect. We used ultrasound to target and find the center of the bowel here, shown in this ultrasound called cross-section. And each one was a planned 2.5 centimeter spherical treatment in the phantom. Because of the gas blockage, the ultrasound image did not show the bubble cloud, which is usually used to verify the presence of cavitation here. So instead, we used the auditory feedback to select the treatment voltage. After treatment, the bowel was inspected for any damage. The diameters were measured inside the phantom of the treatment zone, and there eventually then was a histological evaluation of the bowel. In all cases, treatments were successful. The treatments appeared like this, just a different texture within that phantom. And this includes all instances of partial blockage by gas-filled bowel. When we start to look at the volumes here, we see that the volumes when there was blockage by the gas-filled bowel are larger than the control group. And while this might be statistically significant, where indicated with the asterisk, this might not be clinically significant because these volume differences are small. When we look at the diameters here, a trend starts to emerge. We see that the Y diameter of the treatment zone is wider. This is running perpendicular to the bowel, illustrated in this blue illustration here, while the X and Z axes are roughly unchanged. They have a greater effect when the focal point is closer to the gas-filled bowel, indicating that there could be some sort of shifting or distortion of this that's occurring when more of the beam path is passing through that space. There was no damage observed in any of the bowel. This includes visible damage or air leaks or pneumatosis. When the histopathology was evaluated, any changes that were seen were thought to be due to preservation effects, as these were seen in both the treated and untreated portions due to the ex vivo model. We were limited by this ex vivo model here. Again, those effects that we saw due to the preservation. We plan to retest this with immediate treatment and fixation on the day of sacrifice in the future, and then hopefully plan to move into an in vivo model. We also plan to investigate other blockage scenarios, including completely blocking the path with gas-filled bowel or other structures, as well as liquid-filled and including the bowel itself in the treatment zone. So in conclusion, histotripsy treatments can be made when there's partial blockage of the beam path with gas-filled bowel. This results in a larger volume than would otherwise be made without that blockage. And it appears to be safe to the bowel, though further investigation is needed before this could be moved into humans. Thank you.